This video contains audio content only. There are no visual elements or images accompanying the audio. If you're expecting to see video footage, please note that this is an intentional audio-only presentation. We recommend listening to this content rather than watching the screen. Thank you for your understanding and enjoy the audio. Welcome. Ever wondered why some animal hybrids, like mules, can't have babies? This article dives into the fascinating world of hybrid incompatibility, exploring why some cross-species offspring thrive while others face reproductive dead ends. We'll uncover a curious pattern where one sex is often more affected in these hybrid mixes, a phenomenon known as Haldane's rule. Get ready to explore the genetics of speciation and uncover the evolutionary forces that shape the diversity of life, all explained in easy-to-understand language. Chapter 1. The Curious Case of the Missing Hybrids. Nature has a peculiar way of reminding us that life, even in its most diverse forms, often shares common threads. One of these threads, woven through the tapestry of the animal and plant kingdoms, is the curious phenomenon of hybrid incompatibility. Imagine nature as a grand mixing pot, where the ingredients are different species, and the recipe is hybridization. Sometimes, the resulting concoction is a harmonious blend, a hybrid offspring that thrives. But other times, the mixture turns sour, producing offspring that are far from a perfect blend of their parents. Consider the humble mule, the offspring of a male donkey and a female horse. These sturdy creatures are a testament to the possibility of interspecies breeding, yet they are also a prime example of hybrid sterility. Mules, almost always males, are robust and often possess desirable traits from both parents, excelling in strength and endurance. However, they are incapable of producing offspring of their own, representing a dead end in the reproductive chain. This isn't just a quirk of equine hybridization. Similar stories unfold across a vast array of organisms. Shifting our gaze to the avian world, we encounter a different twist in this hybrid tale. In birds, the female is the one with two different sex chromosomes, and it's often the female hybrids that exhibit sterility or inviability. For instance, consider the intriguing case of Cox's sandpiper, a bird initially thought to be a distinct species. Upon closer examination, however, all known specimens were males. This led scientists to suspect that Cox's sandpiper isn't a species at all, but rather the male offspring of two different sandpiper species, with the female hybrids being either incredibly rare or non-existent. The phenomenon isn't confined to the animal kingdom. Plants, too, play a part in this hybrid drama. Take the Silene genus of flowering plants. Some Silene species, with distinct sexes determined by sex chromosomes, also follow this peculiar pattern. When crossed, their hybrid males are often rare and sterile, mirroring the pattern seen in mammals and fruit flies. These examples, spanning flies, mammals, birds, and plants, illustrate the widespread nature of this hybrid phenomenon. But the story doesn't end with just sterile offspring. Sometimes, the sex bias in hybridization manifests as a skewed sex ratio. One sex might be entirely missing or drastically underrepresented among the hybrid offspring. This imbalance can have profound consequences. If one sex is absent, it creates a reproductive dead end for the hybrid population, even if the other sex is perfectly fertile. It's like having all the ingredients for a cake but no oven to bake it in. This hybrid dysfunction, whether it's sterility or a skewed sex ratio, acts as a reproductive barrier between species. It's a roadblock on the path of gene flow, preventing the complete blending of two distinct gene pools. This barrier plays a crucial role in the process of speciation, the evolutionary journey where one species diverges into two. The observation of these unusual hybrid patterns wasn't a recent discovery. Early naturalists, meticulously documenting the diversity of life, stumbled upon these peculiarities long before the advent of modern genetics. They found these patterns surprising, a deviation from the expected blending of traits in hybrids. Some proposed that the differences in sex chromosomes might hold the key to this mystery. Others speculated about an inherent weakness in one sex when faced with the genetic jumble of hybridization. These initial observations, though lacking the tools of modern genetic analysis, sparked a quest to understand the underlying mechanisms. Why was one sex consistently more vulnerable in hybrid crosses? What genetic factors orchestrated this bias? This curiosity paved the way for a deeper exploration of the genetic basis of hybrid incompatibility. Scientists began to unravel the intricate dance of genes, the interactions that could lead to sterility or inviability in hybrid offspring. This journey of discovery, fueled by observation and experimentation, ultimately led to the formulation of a principle that would shed light on the curious case of the missing hybrids. Haldane's rule. Chapter 2. The heterogametic riddle. Why one sex suffers more in the hybrid world. In 1922, British biologist J.B.S. Haldane, while studying hybrid crosses between different animal races, stumbled upon a curious pattern. He noticed that when one sex was conspicuously absent, rare, or sterile among the hybrid offspring, it was almost invariably the heterogametic sex. This observation, initially documented in a paper titled Sex Ratio and Unisexual Sterility in Hybrid Animals, marked a groundbreaking discovery in the field of evolutionary biology. Haldane's observation wasn't just a peculiarity of a single species. It proved to be a recurring theme across diverse animal groups. He initially noted its presence in mammals, where males, with their XY chromosomes, are the heterogametic sex. Further investigations revealed the same pattern in lepidopterans, the order encompassing butterflies and moths, as well as in birds, orthopterans, grasshoppers and crickets, and dipterans, flies like Drosophila. This widespread consistency hinted at a fundamental principle governing hybrid crosses. But what exactly does heterogametic mean? The term refers to the sex possessing two different sex chromosomes. In mammals, this is the male, carrying an X and a Y chromosome. Females, with their two X chromosomes, are termed homogametic. In birds and butterflies, however, the roles are reversed. Females are heterogametic, denoted by ZW chromosomes, while males are homogametic with ZZ. Other variations exist, such as XO systems where one sex has only a single X chromosome, and ZO systems where one sex has only a single Z chromosome. The crucial point is that Haldane's rule applies regardless of which sex is heterogametic. Whether it's the XY males of mammals or the ZW females of birds, the heterogametic sex bears the brunt of hybrid incompatibility. Hybrid dysfunction, the umbrella term for the problems plaguing hybrid offspring, manifests primarily as sterility and inviability. Sterility, in this context, signifies the inability of hybrids to produce offspring, rendering them reproductive dead ends. Inviability, on the other hand, encompasses a range of developmental issues, from reduced survival rates to embryonic lethality. Hybrids might fail to develop properly, be born with debilitating defects, or simply not survive to reproductive age. Haldane's rule becomes particularly relevant in the early stages of speciation. As two populations begin to diverge genetically, they might still be able to interbreed, producing hybrid offspring. However, these hybrids, especially those of the heterogametic sex, often suffer from sterility or inviability. This hybrid dysfunction acts as a critical barrier to gene flow between the diverging populations. It's like a dam holding back the mixing of two rivers. This barrier is crucial for reproductive isolation, the cornerstone of speciation. Without this isolation, the two populations would continue to interbreed, their genetic differences would blur, and the formation of new species would be thwarted. Haldane's rule, therefore, isn't merely a biological curiosity. It's a fundamental principle driving the very process of speciation. Haldane's rule also applies to scenarios where two species, having diverged in isolation, allopatric speciation, come back into contact, secondary contact. If they begin to hybridize in a shared area, sympatry, Haldane's rule still holds. The heterogametic sex of these hybrid offspring will be more likely to experience sterility or inviability. This pattern has been documented in a variety of organisms. For instance, two species of howler monkeys, Alwata caraya and Alwata chlamydens, have been observed forming mixed groups and producing hybrid offspring, providing real-world evidence of Haldane's rule in action. In the fruit fly genus Drosophila, extensive research on interspecific hybridizations has solidified Haldane's rule. In a study encompassing over 140 crosses, nearly every instance where hybrids displayed sex-biased sterility, it was the XY males that were sterile, while the XX females remained fertile. This stark contrast underscores the strong influence of heterogameity on hybrid outcomes. Similarly, in birds and butterflies, where females are the heterogametic sex, hybrid females are the ones more frequently affected by sterility or inviability. This reversal of the pattern seen in mammals and flies reinforces the universality of Haldane's rule, emphasizing that it's the heterogametic sex, regardless of whether it's male or female, that is more susceptible to hybrid dysfunction. Even in the plant kingdom, Haldane's rule finds its application. Studies on plants with sex chromosomes, like certain species of campions, have revealed a similar pattern. When different Selene species are crossed, the resulting hybrid males are often both rare and sterile, mirroring the pattern observed in mammals. This extension of Haldane's rule to the plant kingdom highlights its broad significance in the evolutionary dynamics of diverse life forms. Another example involves hybridization between two water strider species, where mitochondrial introgression is restricted compared to nuclear markers, again highlighting the complex interplay of genetic factors in hybrid zones. Studies on newts, pigeons, doves, and butterflies have further corroborated Haldane's rule, showcasing its prevalence across the animal kingdom. Haldane's rule, therefore, isn't just a quirky observation, it's a fundamental principle underlying the process of speciation. It's a testament to the intricate dance of genes, where even seemingly small differences in sex chromosomes can have profound consequences for hybrid offspring. However, the precise genetic mechanisms responsible for Haldane's rule remain a subject of ongoing research and debate. Several hypotheses have been proposed, each attempting to explain the rule's pervasive influence. The dominance theory suggests that recessive alleles on the X chromosome are more likely to be expressed in the heterogametic sex, leading to incompatibility issues. The faster male hypothesis posits that genes involved in male reproduction evolve more rapidly, increasing the likelihood of incompatibilities in hybrid males. The faster X theory proposes a similar rapid evolution for X-linked genes, while the meiotic drive hypothesis suggests that selfish genetic elements can disrupt the normal segregation of sex chromosomes in hybrids. These hypotheses and their intricate interplay will be explored in greater detail in the chapters to come. Unraveling these genetic mechanisms is crucial not only for understanding Haldane's rule but also for gaining deeper insights into the very process by which new species arise. Chapter 3. The X-Files of Evolution. Unmasking the secrets of the X chromosome. But why this bias? The X chromosome, a unique player in the genetic orchestra of sex determination, holds many clues. The dominance theory offers a compelling explanation, focusing on the hemizygosity of the X chromosome in the heterogametic sex. In mammals, males are XY, possessing a single X, while females are XX. This difference in dosage, 1X versus 2, becomes crucial in hybrids. Imagine a recessive allele lurking on the X chromosome of one species, an allele that causes incompatibility with the genes of another species. In a homogametic hybrid female, XX, the presence of a second, normal, X chromosome can mask this incompatibility, like a backup singer covering for a flubbed note. However, in a heterogametic hybrid male, XY, with his solo X performance, there's no backup. The incompatibility takes center stage, potentially leading to sterility or inviability. This scenario mirrors the inheritance of sex-linked traits like color blindness or hemophilia in humans. These conditions, caused by recessive alleles on the X chromosome, are far more common in males. A male needs only one copy of the recessive allele to express the trait, while a female needs two. The heterogametic sex, lacking a second X chromosome to mask recessive alleles, is more vulnerable. Adding another layer to this complexity is the faster X hypothesis. This theory proposes that genes on the X chromosome evolve at a faster pace than those on autosomes. Several factors can contribute to this accelerated evolution. Sexual selection, the competition for mates, can drive rapid changes in genes involved in male traits, many of which reside on the X chromosome. Furthermore, differences in mutation rates between the sexes, with males often exhibiting higher rates, can contribute to faster X evolution. This rapid evolution can lead to a greater accumulation of incompatible alleles on the X chromosome, further exacerbating hybrid dysfunction in the heterogametic sex. The story, however, isn't solely about recessive incompatibilities. Dosage compensation, a mechanism ensuring equal expression of X-linked genes in both sexes, plays a significant role. In many species, dosage compensation involves increasing the activity of the single X in males or decreasing the activity of one of the two X's in females. This delicate balance is often disrupted in hybrids. Imagine two species, each with its own finely tuned dosage compensation mechanism. When these species hybridize, their mechanisms might clash. The hybrid's attempt to balance gene expression can go awry, leading to an overexpression or underexpression of crucial X-linked genes. This imbalance can disrupt development, causing sterility or even inviability. The size of the X chromosome also matters. Studies on Drosophila have revealed a large X effect. Species with larger X chromosomes carrying a greater proportion of the genome tend to exhibit more severe hybrid dysfunction. This makes intuitive sense. A larger X means more genes, and more genes mean more opportunities for incompatibilities to arise. The intricacies of the dominance theory are further illustrated by experiments involving Drosophila with unbalanced genomes. Researchers created female flies carrying two X chromosomes from the same species, but with autosomes from different species. While these females sometimes exhibited hybrid inviability, they often remained fertile. This finding, initially puzzling, suggests that the X chromosome's role in hybrid sterility isn't solely about recessive incompatibilities. The interaction between X-linked genes and autosomes, crucial for fertility, appears to be less sensitive to disruption than the interactions governing viability. This nuance highlights the complex interplay of genetic factors contributing to Haldane's rule. The faster X hypothesis, intertwined with the dominance theory, provides a more complete picture. The rapid evolution of X-linked genes, driven by sexual selection and other factors, fuels the accumulation of incompatible alleles. These alleles, while often recessive and masked in homogametic hybrids, are fully exposed in the homozygous heterogametic sex. This double whammy, a higher likelihood of incompatibilities and their full expression, makes the heterogametic sex particularly susceptible to hybrid dysfunction. It's the outcome of a complex interplay of genetic factors, with the X chromosome playing a starring role. The homozygosity of the X in the heterogametic sex, coupled with its accelerated evolution and the intricacies of dosage compensation, creates a perfect storm for hybrid incompatibility. This understanding, gleaned from decades of research on diverse organisms, from fruit flies to mammals to plants, underscores the X chromosome's pivotal role in the evolutionary drama of speciation. Chapter 4. Beyond the X. Meiotic drive and the race for reproductive success. The X chromosome, with its homozygosity and faster evolutionary pace, undoubtedly plays a starring role in the drama of hybrid breakdown. However, the story isn't solely an X chromosome narrative. Other evolutionary forces, particularly meiotic drive and the faster male theory, contribute to this complex interplay, especially regarding hybrid sterility. Meiotic drive, a phenomenon where selfish genetic elements manipulate the gamete formation process, adds a layer of intrigue to this evolutionary tale. These genetic elements, often located on sex chromosomes, act like saboteurs, biasing the transmission of their own copies to the next generation, even at the expense of their host's overall fitness. Imagine an X-linked gene in Drosophila that produces a toxin harmful to Y-bearing sperm. This toxin, while potentially detrimental to the male's overall fertility, ensures the survival and propagation of the driving X chromosome. In a normal population, this drive can lead to skewed sex ratios, with fewer males being produced. However, within a species, suppressors often evolve to counteract these drive elements, restoring a more balanced sex ratio. This delicate balance is disrupted in hybrid populations. When two species with different drive systems hybridize, the suppressors from one species may not effectively counteract the drive elements from the other. This can unleash the full force of the drive, leading to severe sex ratio distortions and sterility in the heterogametic sex. For instance, if the X-linked drive from one Drosophila species is unchecked by the suppressors of another species in a hybrid male, it can decimate Y-bearing sperm, rendering the male sterile. This scenario highlights how a trait that provides an evolutionary advantage within a species can become a liability in a hybrid context. The hybrid genome becomes a battleground for competing drive systems, leading to reproductive chaos. The faster male theory offers another perspective on the evolution of hybrid sterility. In many species, males face intense competition for mates, a driving force behind the rapid evolution of male reproductive traits. This rapid evolution, fueled by sexual selection, can lead to significant divergence in male reproductive genes between closely related species. Consider Asian elephants, where studies have revealed a significant reduction in male-driven gene flow, suggesting faster evolution of male traits. This rapid divergence can create incompatibilities when these species hybridize. The intricate machinery of male reproduction, honed by generations of sexual selection, might not mesh well with the corresponding machinery of a different species. This mismatch can manifest as reduced fertility or even complete sterility in male hybrids. For instance, proteins essential for sperm function in one species might not interact correctly with their counterparts in another, leading to dysfunctional sperm and male sterility. However, the faster male theory, while effective in explaining male sterility in male heterogametic species, faces challenges when applied to species with female heterogamety. In birds and butterflies, where females are the heterogametic sex, the faster male theory would predict male sterility in hybrids. Yet, in many such cases, it's the females that exhibit reduced fertility. This discrepancy suggests that other factors, such as dominance or faster Z evolution, are likely at play in these systems. In certain Drosophila species, where females are heterogametic due to maternal effects, exceptions to Haldane's rule have been observed, further highlighting the complexity of these interactions. The evolution of hybrid dysfunction, therefore, is a multifaceted process, influenced by a complex interplay of genetic factors. Dominance, meiotic drive, and the faster male theory, while often treated as separate explanations, can interact in intricate ways. For example, the faster male effect can exacerbate the impact of recessive incompatibilities on the X chromosome in male heterogametic species. Furthermore, meiotic drive can interact with both dominance and faster sex chromosome evolution to produce complex patterns of sterility and sex ratio distortion in hybrids. In the hybrid world, it's a battle for survival at the genetic level. The fate of a species can hinge on the intricate interactions of its sex chromosomes, the presence of selfish drive elements, and the evolutionary pressures exerted by sexual selection. The dominance theory provides a foundation for understanding the role of recessive incompatibilities, while the faster male theory highlights the importance of sexual selection in driving the divergence of male reproductive genes. Meiotic drive adds another layer of complexity, with selfish genetic elements battling for supremacy in the hybrid genome. These forces, acting in concert or in opposition, shape the complex landscape of hybrid fitness, determining whether hybrids thrive, struggle, or simply fade away. The study of Haldane's rule, therefore, provides a window into the intricate mechanisms of speciation, revealing the genetic battles that shape the diversity of life. Instances where multiple factors likely interact. In some Drosophila species, while dominance theory predicts sterility in females with unbalanced genomes, two X chromosomes from the same species, autosomes from different species, these females often remain fertile, suggesting interactions with faster X or meiotic drive are at play. In marsupials, where both sexes are functionally homozygous for the X chromosome due to paternal X inactivation, the dominance theory's explanation of Haldane's rule is challenged, indicating the potential involvement of faster X or faster male effects. In Asian elephants, reduced male-driven gene flow suggests faster male evolution. However, the interplay with dominance effects on sex chromosomes needs further investigation to fully explain hybrid dysfunction. In water striders, restricted mitochondrial introgression compared to nuclear markers highlights potential interactions between cytoplasmic factors and sex-linked genes in shaping hybrid zones, possibly involving meiotic drive or dominance. The evolution of XYY sex chromosome systems in some saline species contributes to male rarity in hybrids, interacting with dominance effects on sterility. This suggests a complex interplay of chromosomal rearrangements and genetic incompatibilities in plant hybrid breakdown, potentially involving faster X or meiotic drive. These examples illustrate how the forces of dominance, meiotic drive, and faster sex chromosome evolution can interact in complex and often unpredictable ways, shaping the intricate landscape of hybrid fitness and the ongoing process of speciation. Chapter 5. Beyond Chromosomes. Exploring the unexpected frontiers of Haldane's rule. Haldane's rule, the observation that the heterogametic sex often bears the brunt of hybrid incompatibility, extends beyond the familiar narrative of sex chromosomes. While differences in sex chromosomes play a significant role, a deeper exploration reveals a surprising cast of characters influencing the fate of hybrid offspring. Cytoplasmic elements, enigmatic B chromosomes, and the rapid pace of male evolution all contribute to this intricate dance of speciation. The cytoplasm harbors its own genetic material, primarily within mitochondria. These maternally inherited cytoplasmic genes create a unique dynamic in hybrid crosses. A mismatch between maternal cytoplasm and a blended nuclear genome can lead to cytoplasmic incompatibility, disrupting cellular functions and reducing hybrid fitness. In the tiny copod Tigriopus californicus, for instance, interpopulation hybrids exhibit disruptions in mitochondrial function. The maternal inheritance pattern of mitochondrial DNA creates an evolutionary asymmetry, potentially leading to incompatibility with the nuclear genome of a different species or population. This incompatibility can manifest in reduced fertility or developmental abnormalities, contributing to the sex bias observed in Haldane's rule. Adding another layer of complexity are B chromosomes, extra chromosomes found in some individuals. These enigmatic elements don't follow the usual rules of inheritance and can have surprising effects on hybrid fitness. The evolutionary pressures shaping the sexes don't stop at the cellular level. The interplay of these different evolutionary forces creates a complex landscape for hybrid fitness. The composite hypothesis suggests that different subdivisions of Haldane's rule, like sterility versus inviability, might have distinct underlying causes. In Drosophila, for instance, XY interactions are crucial for hybrid sterility, while in marsupials, both sexes experience similar incompatibilities due to paternal X inactivation. These differences highlight the diverse genetic mechanisms contributing to Haldane's rule across different phylogenetic groups. Adding another layer of complexity is the observation of within-species variation for Haldane's rule. The severity of hybrid dysfunction can vary between populations of the same species, suggesting that the genetic basis of Haldane's rule isn't fixed. This variation can provide valuable clues for distinguishing between different contributing mechanisms. Furthermore, considering sex differences in heterosis at shorter genetic distances can provide a link to understanding sex-biased hybrid dysfunction at greater genetic distances. Heterosis refers to the phenomenon where hybrids exhibit increased fitness compared to their parent species. However, this increased fitness is often sex-specific. These patterns of heterosis at shorter genetic distances can offer insights into the potential for sex-biased hybrid dysfunction as genetic divergence increases. Haldane's rule, therefore, is not a simple, monolithic principle. It's a multifaceted phenomenon, shaped by a complex interplay of genetic and evolutionary forces. From the microscopic battles within cells between cytoplasmic and nuclear genes to the grand sweep of sexual selection driving rapid male evolution, these diverse factors contribute to the intricate dance of speciation. By studying the various mechanisms contributing to hybrid dysfunction, we gain valuable insights into the evolutionary forces shaping biodiversity. Haldane's rule has become a powerful tool for understanding the evolutionary tree, revealing the genetic pathways that lead to the formation of new species. Haldane's rule reveals that the heterogametic sex, XY or ZW, is more likely to experience sterility or inviability in hybrid offspring. This seemingly simple observation has profound implications for understanding speciation, highlighting the role of sex chromosomes, cytoplasmic incompatibility, and rapid evolution of male traits. From fruit flies to elephants, the widespread prevalence of this rule underscores its importance in evolutionary biology. These findings offer a deeper understanding of the genetic barriers that maintain species boundaries and the complex interplay of factors driving biodiversity. Where do you think research on hybrid dysfunction will lead us next? Share your thoughts and questions below. Don't forget to like, subscribe, and suggest topics for future explorations. Keep questioning, keep learning, the wonders of evolution await.